My name's Lindsey VanSandt. I am the director of the Imperiled Cat Signature Project at the Center for Conservation and Research of Endangered Wildlife, or CREW for short, which is in the Cincinnati Zoo and Botanical Garden.
Hello, I'm John Rossi. I'm a touring drummer with a passion for animal conservation. When I'm on the road, I spend as much time as possible visiting zoos, aquariums, and conservation organizations. Now, I want to share those places with you. I'll be talking to keepers, vets, conservationists, anyone who can help me in my mission of connecting my people to animals through their people. Join me on my Rossifari. Hi, hello, how are you? Welcome back to a very quip-filled episode of the Rossifari podcast. So, as you will hear in the interview, this episode has been a long time coming. I met Dr. VanSandt a few years ago, and we have wanted to make this episode happen ever since, and we finally did. And Lindsey is honestly one of the funniest, most natural, entertaining guests I've ever had on the podcast. The stories are going to crack you up. I think I came up with something like 7 titles for this episode, when often I struggle to come up with one. And they're all just quotes, most of which were from Lindsey. And then I actually put my 4 favorite up for vote on our Discord. So that's also your friendly reminder that, hey, we have a Discord. So the title that won at the vote there is the title that you see. And the vote's still running as I record this, so I don't know what it is yet. But yeah, so looking forward to sharing. This is just such, Lindsey has such a cool story, y'all. It's so cool. The work being done is so cool. Her actual story is so cool. It's going to blow your mind. So we will get to that in a minute. But before we do, make sure that you hit subscribe or whatever app you're using, whatever thing like subscribe there is, so that you never miss an episode of the podcast. Make sure you're following along on the socials at Rossifari, at Rossifari Pod on the TikTok machine. You can check the show notes for our link to the Discord, and you can join the Rossifari community there. And don't forget, there's a Patreon to support the pod for as little as $3 a month. You can help support me. Yay! So with all of that said, let's get to it. Here's my interview with Dr. Lindsey VanSandt of CREW at the Cincinnati Zoo. Yes, so many words,
so many
and names. So we met, goodness, a couple years ago, 3 years ago now, I think, at the AAZV conference. Yeah. And I instantly knew that you would be a great podcast guest because you are a firecracker. And we got permission to do it, and the conference was too busy. And then we were like, cool, we'll catch it next year at the conference. And the next conference was too busy. And then I haven't been back to Cincy until now, which is crazy. Wild. I used to come once or twice a year, but life has just taken me in different directions. So here we are, 3 years later. I'm so excited. Yeah. So let's start off before we get into what this actual project that you work on is. Mm-hmm. How did you get here? What was your career? What were your passions growing up? All those things.
Yeah. I'm from Missouri, and I will say it started with my mom was like the original conservationist. Like, before, back in the day when you had to drive your recycling to a place and pay for it, my mom was the one with the can crusher in the garage.
Oh my God.
Yelling at everybody else. We had compost in the back. So I feel like I always had this strong interest in conservation. I always wanted to be a veterinarian. I would go to zoos, and it's funny because I was a kid, I didn't understand. I would always like the nursery and people bottle feeding whatever baby. And I thought, that's what I want to be. I thought I was going to be a zoo vet. I was in vet school, had my first opportunity to work at a zoo. I actually went all the way to Johannesburg to work there at the South Africa Johannesburg Zoo. And one day into a 6-week externship was like, I don't want to do this. I'm very mammal centric, and we don't. Most vet schools, Missouri included, you don't get a lot of exotics. Mm-hmm. Domestics are great models for a lot of them, but they're all mammals.
Yes.
And like the first day, they're bringing in this lizard, and I'm like, I have no idea what this is, And I thought, oh my gosh, when I go, I love going to the herp house when I go to a zoo, but I don't retain any long-term knowledge. I'm like, I'm going to be a terrible zoo vet. Somebody brought in a turtle, and I was trying to intubate it. And I look everyone around me and I go, because I have my hand in its mouth, and I'm like, do turtles have teeth?
No.
And they're like, I'm like, I literally have no idea what I'm doing.
Oh, no.
This is not the place for me. So I was good. It's like I always tell people, whatever you think you want to do, go do it because you may be very wrong.
Yeah.
So I came back to vet school. I was like, my life is a lie. I was president of our Zoo and Exotic Club.
No.
And everyone's like, how was it? You got to go to South Africa. You got to work at a zoo. And I was like, it was fine. It's fine. But I'd actually also worked in a lab in undergrad, kind of checking that box. I didn't really see myself as a scientist, but checking that box for like something that they like to see. Like, you know, you kind of look at what applicants want and they really like that. And I worked in a PCR lab and there was just something so magical. I really like the tedious carefulness of like benchtop work, but then this magical thing happens and PCR and, you know, all polymerase chain reaction. And then there's this band and it tells you this information. So I thought it was cool. But then it was actually Dave Wilt, who was the director of what we call now the Smithsonian Conservation Biology Institute, came and gave a talk about wildlife reproduction research. And I also really love reproduction in undergrad, but in Missouri, unless you're either, you know, going to people who do the breeding are usually those backyard breeders that don't come to vets. And I didn't really want to do that with my life. I didn't, you know, dairy cows. Okay. But so I thought, well, reproduction's really cool, but it's not what I'm going to do. So then this person came and I realized there's this little niche world that's like perfect for me. So that was helpful. And then I kept in contact with him. I actually ended up at the St. Louis Zoo for a year after that, on the research side.
So did you finish vet school?
Oh, yes.
Okay,
yes,
so you
sorry.
did decide to go through it. Okay.
Yeah, although here's what's hilarious is I actually never got a bachelor's degree. I applied.
How?
You don't have to for a lot of vet schools.
No, but getting in is so competitive.
Yeah. Well, I actually didn't think I'd get in my first year. So I applied and I was animal sciences and I loved a lot of that coursework. But I started looking at the coursework in vet school because I had one of the best advice I got from somebody in vet school was take as many classes as you can that will mirror your first year of vet school because it's so much information. And a lot of the classes I wanted were biology, not animal sciences. So I sort of low-key changed majors without actually changing majors. And so I ended up taking a bunch of these like capstone classes, you know, and but it wasn't even in my major. But they were the classes I needed.
Right.
And so then I realized, like, I don't have enough of either. I have all these things. And the irony is I almost could have made the biology work, but I'd taken Spanish in high school and I had 2 years of Spanish, but they wanted 3 years of Spanish for biology, which is cool, or 3 years of a foreign language. And then another irony, I'd gone to Costa Rica for a summer to live actually hilariously on a dairy farm and learn about sustainable agriculture. And I didn't pay extra for the language credit because I was like, I just want to go and do this thing.
Oh.
So I say all of this because I'm about to graduate from vet school and I don't have, I've been in college 8 years. And when I'm filling out applications, highest level of education complete, high school.
Oh my God.
So yeah, I'm glad I finished vet school because that would have been a bummer.
That's amazing. Did I mention I knew you'd be a great interview?
So then I went to St. Louis. It was actually an unpaid internship, as many of them were back in the day. I'm happy now to know that that same experience exists, but now it is paid.
Nice.
But it worked out because it meant I had to use my vet degree and work at an emergency clinic. So it was really useful to actually keep all those vet skills up.
Sure.
Otherwise, I wasn't using them as much because, again, I was on the research side of the St. Louis Zoo, not on the vet side. And so then after that, I ended up at, kept in contact with Dave Wilt, and then a kind of weird world, other connections. It's kind of a funny world we live in. How this person helped me with that, this, that, or the other. I ended up interviewing with a researcher there, Dr. Buddha. I have known him now, oh my gosh, it's 2009, 15 years, and I still can't say his last name correctly. Pukudenti. He is somebody who used to do a lot of cat research. And then he had completely switched into hoofstock. Like a fool. I don't do any current research on what he's up to. I just know him as like the cat sperm guy. And I love cats. And I'm like, this is perfect. Now, my experience with vet school is I actually worked in the equine ICU as a tech. And again, growing up in Missouri, I loved most of the reproduction or most of the work I did was at mixed animals. So I have all this hoofstock experience. He's looking at my CV like, oh, it's a hoofstock person. I'm thinking he's the cat guy. And I don't think there's ever been an interview in the history of the world with more mutual disappointment in the other one of like, oh, you want to do oh, you do hoofstock now for a very long time, very searchable on the internet. I know this guy, no problem. So that went great. But he invited me out to kind of meet everybody and kind of do an interview. And I realized, getting a PhD is about learning how to do research. The species itself doesn't really matter. It's how to do the research. And I like hoofstock. That's fine. They're still a mammal.
Right. Yes, they
They're
are.
still checking that box for me. So I was like, fine. And then, so my PhD was you can't get a degree, obviously, with like a zoo. So it was actually at the University of Maryland College Park. And I did zero, yet again, failures of past Lindsey, zero recon on the people there. I was sort of randomly assigned an advisor, but I didn't really care because I thought I was going to be at Smithsonian and like that was just the degree granting institution.
Right.
And then we couldn't get funding. And I was like, well, if I can't get funding, I might as well do something I want to do. So I completely flipped like what my project was from working on like the Dama gazelle and just kind of doing a lot of characterization work. And I should mention this too, my advisor at Maryland, Dr. Carol Kiefer, who I adore, she's like a dear friend and colleague to this day. So I'm so grateful that I just randomly got this person. Her background is in like stem cells and cloning and more, like molecular techniques. And so I came up with this idea of looking at, I wasn't the first one to think of this, but spermatogonial stem cells are these stem cells that are in testes and they just make sperm. So unlike other stem cells that, do a lot of stuff, they're called unipotent. They just make sperm. But unlike a mature sperm cell that can't replicate, right? The only way it's going to reproduce is finding an egg and making a new individual. These stem cells can replicate. So it's like an alternative source of genetic material. So this work had been done largely in rodents. And I was like, let's do it in cats. And so I still had a PhD that was in cat repro, although again, it was more from a conservation standpoint. It was conservation in mind of like, this could be a good model. If we could get, when an animal dies, instead of just trying to collect some of their sperm, we could get their stem cells. And I had all sorts of crazy ideas. You could actually, in rodents, you can transplant the stem cells from from a rat, like from one individual into another individual. And those stem cells will actually start making sperm.
Oh my gosh.
And so, yeah, which is wild. And so the idea was cats are so closely related. Imagine if when a tiger dies, I can get his stem cells and then transplant them into a domestic cat. And then I have all these domestic cats making all these like super endangered sperm, train them for voluntary semen collection. And we just have this like sperm farm of all these really important individuals.
Oh my God.
So that was the dream.
That's an incredible, like just whether or not it works is irrelevant. Just thinking that way is incredible. Like that's so outside the box.
Well, again, the spermatogonial stem cell field existed before me. I just was like, we should do this in cats. But it really just meant that I drove to a lot of spay-neuter clinics and they called me the gonad girl. Gonad girl's here. Because the first time I was trying, there's people in the room and I'm just here. I'm like, hi, I'm here to pick up, I'm trying to be subtle because there's people here to pick up their cats. I'm here to pick up the gonads. And of course, the lady at the front desk doesn't know what that means.
Right.
And she's like, the what? And I'm like, gonads.
We don't have a patient here named
right,
that.
Right. And then I'm like, the testicles? And she's like, what? You want the testicles? And I'm like, oh my God. And so after that, they're like, gonad girls here. And then they would just hand me my cooler and then I would go and do all of the lab work. But yeah, we got pretty far on it. We could culture them, which was cool. I actually took that research with some of it with me here. We actually tried to do transplants from my stem cells into some of our males here. But we realized the technique is really hard. When you're doing this and they were doing it in bulls and pigs. Those are really big animals.
Yeah.
You can actually get to the testis and see it. In really small animals, it's kind of like a micro dissection, like in rodents. And cats are somewhere in the middle where we couldn't access it the same way you can in rodents. So we were trying to do ultrasound guided.
Oh my gosh.
And these are 3-month-old cats.
Oh no.
So yeah, all of your listeners should just have an opportunity to look at kitten testicles and try to imagine putting an ultrasound probe on that and then getting the needle exactly where it went. So we think one of them, we had a little bit evidence, but that was it. But that's the end of that part of my life. I don't do any work in that anymore. So it sort of worked.
That's sort of amazing.
Yeah.
I think that might be the single most convoluted story to getting somewhere that I've heard on this podcast. So.
That's my superhero power is convoluted answers.
I like that. I mean, I'm a conservationist by way of being a drummer. So right, I get it,
Yeah.
But Wow. Okay. So how did you find the project that you're working on now? How did you get involved here at Crew? All of that stuff.
Good question. So actually, I also have Dr. Aaron Curry here to thank for that, who directs our polar bear, or I believe now it's just bear signature project.
Aaron's awesome.
Then in a weird small world, somebody I was in grad school with at Maryland, Aaron had gone to undergraduate with, and they were really good friends. And the magic of social media, me and Aaron were at some reproduction conference. We both had posted about it, and our mutual friend was like, you two should meet. And so then I'm talking to her, and she's like, oh, you're a vet? You're getting a PhD? You want to do cat work? You know, and like my former boss, Bill Swanson, who's actually officially retired as of today.
Oh.
Yeah.
Nice.
He was looking for a postdoc and wanted a veterinarian because we have a domestic cat colony here, but wanted research background and all of that. So it was really magical timing of when I was finishing up with everything. He actually had me come out for like sort of an impromptu interview, and he invited me out. It was like 2 weeks before my thesis was due, but people from Smithsonian were coming out here to learn a technique. He's like, just come with them, and then you can kind of do an interview. And so I'm in the car, like writing on my laptop.
Gosh.
And this is the spermatogonial stem cell stuff. There's all these like immunohistochemistry images. So it's like fluorescent images. So it's really hard to see on a laptop in the day. So I have like this towel I brought. So my laptop, I'm just in the back with a towel over me so I can like be in the dark. I was up all night writing a chapter, like getting edits back. And so it was the most calm interview I've ever. Job interviews make me so nervous. I lose all my personality. I don't know my name. Like, I don't like doing job interviews. So it was perfect, though. I was so tired. I had no cortisol left to give. And it was very relaxed and it was fine. And I was like, wow, this is great. So at some point we have I mentioned the domestic cat colony. So I was helping with the anesthesia on the cats while they were learning how to do this artificial insemination technique. And Bill's like, how are we doing over there? And I said, I'm great, Bill. Thanks for asking. He's like, and the cat? I'm like, oh, the cat. She's good too. We're fine. You guys keep going. Everything's great. So that worked out. I got a phone call while I'm standing in line to walk for graduation when he popped the question to offer me a job, which was great because people always ask you at graduation, what's next?
Right.
And I'm like, I have an answer. So that really worked out well. I honestly thought I even if I could just be in reproduction, you know, I kind of made my peace with that starting my PhD. I thought everybody wants to do cats. They're the best. I'm happy with hoofstock. I'm happy with whatever. St. Louis was mostly canids. I'm like, they're kind of like cats. That's fine. I'm happy. And so the fact that I now get to primarily work on cats is like, just blows my mind and brings me great joy.
That's awesome. I love that. That's so cool. So what do you do with cats here? Yes.
What do I do? Okay. So we focus on two main areas. The first is developing what we call assisted reproductive technologies for cats, or ART. So it's very similar to what you hear about in humans, things like in vitro fertilization, where you're taking sperm from a male, eggs from a female, putting them together. I love word origins, by the way. I love in vitro. It literally means in glass, because back in the day, all lab dishes were glass.
Right,
so in vitro fertilization just means fertilizing in glass.
That's really cool. Did not know that.
So like developing those kind of techniques, or artificial insemination where you're collecting sperm from the male and then putting it into the reproductive tract of the female. We also do a lot of work where we're more hands-off. It's just helping determine when a cat's in estrus, helping determine when to put them together, helping determine if pairs are together and they're not successfully reproducing, why. So it's nice that myself and then another member of my team are both veterinarians. And so we've really worked on reproductive evaluations. What can we do? And especially with females have been a really black box for us. Males, it's kind of fun because you get to collect sperm, and unlike the stem cells I worked with in college, they move, they swim. So it's really great because you get this instant gratification. And there's a lot of tests we can do on how, even sperm that's swimming may not be able to fertilize. But the female historically has really been a black box for us because there's not as much we can do. We don't typically just collect the eggs and then see if they fertilize because it's more invasive. So just working on the veterinary side of what additional tests, what diagnostics can we do, and how do we just determine what's normal for these cats, which is a big question. So that's a big piece of it. We serve as a lot of myself and members of my team. We serve as reproductive advisors for various SSPs. There's actually, fortunately, a sand cat that was just euthanized yesterday. But we have her ovaries downstairs right now, getting them prepped to do in vitro fertilization. So that's kind of the sad part of our job, but a nice part too, that we do a lot of what we call those gamete rescues. So especially animals that may not have a chance to reproduce while they're alive, it's really important to preserve those genetics. And so we've been developing better ways to do that for both the males and the females.
Interesting. Okay. So what will happen with those eggs? Like, let's say everything works.
Yeah.
Right?
So
talk me kind of through that.
Sure. So right now, Amy, my research associate, Amy Miller, is down there. She's probably done with it by now, but we basically dice up the ovaries. And then from there, we get these eggs or oocytes. But the problem is they're not mature. And so then they have to go in the dish. So they're actually in vitro maturation happens first. And so they'll be in this media for about a day where they mature. And then they're capable of being fertilized. And so luckily, because we have a very extensive sperm bank, we have plenty of males to choose from. We're also really lucky that we have, for the Sandcat SSP, we have somebody that advises us on the genetic side. So they've already created for us this wonderful table that has all the males in the population, all the females in the population. I love it. It's called Mate RX, like Matrix, but Mate R. It's so, I don't know who came up with that, but give them a raise. And then it scores every potential pairing. But they also have included, it used to just be all the living animals, but because we have a sperm bank, they've included all the sperm we have banked.
Oh, wow.
So we can just go in there, we have this beautiful thing, and we can just look at who's a good pairing. And we do a lot of post-analyses, so we know this male has really good sperm. So I believe this female is getting Sven. But so her eggs are going to mature through tomorrow. And then tomorrow they'll move into the in vitro fertilization medium, and then we'll thaw that sperm, put it in there, let that sit for a couple days. Hopefully then what we call cleavage, which it's reproduction, but not that kind of cleavage. It's where how we know the cell fertilizes. You can see the nucleus, and one cell becomes two becomes four becomes eight. So as soon as we see that, we then freeze those embryos. And then it's just a question of having a good potential recipient in the future. The good news is that technology has worked. We've had success with embryo transfer in the past. So often the female that we would choose is probably a proven female, so that we know, like, reproductively healthy and all of that good stuff. But then also that means her genetics are already pretty well represented in the population. And so that way we can sort of have a really good use for her, but not keep breeding her because she's already she's already well represented. So that's a typical candidate we would use for something like that.
Interesting. That's very cool.
Yeah,
I like that. That's really interesting. So does the dead cat SSP count? Like, is it all one thing? So you've got the sperm, you've got the egg, you've got the past, like the animals that have passed, and they're all in there. Is that still just kind of part of the regular SSP? Could you have sperm from a live sand cat go into those eggs, or
absolutely.
Like it's all one.
Oh yes,
okay,
yes,
cool,
yeah.
Okay.
Yeah, it's all one population. We just frozen because we don't have like we will when we're thinking more about artificial inseminations, which obviously tend to be more planned procedures. We can use frozen or fresh sperm for that as well. Usually fresh is if it's a pair that doesn't want to be together for whatever reason, behaviorally or otherwise. Or sometimes there's just a really good match, but he's 3 hours away.
Sure.
So rather than transfer him to a new zoo, we just go collect him, go on our little happy road trip, which we've done many of those, and then do the procedure there. It's just we don't have a sandcat male here. And if he did, we wouldn't know if he'd be a good match. And then also getting everybody on board, because we just got the phone call yesterday at 3:00 to be like, hey, can we knock down our male in 12 hours and get sperm? So that's what the frozen sperm is really nice to give us that flexibility.
Yeah, that's really cool.
It's all under one population. Wow. And I should say this too, it's not just AZA. Like, we work with the European zoo groups. And so we actually do have founder sperm here, not just from there, but like there's a zoo in the UAE that we actually have collected sperm from their sandcats that's in our bank. So all of those are founders for this population. So we try to think about this, a lot of our populations, not even just within our sort of North American zoos, but trying to have a more world management of these cats.
Makes sense. I would think even outside of cats, just in general, that the idea of being able to, we talk about the SSPs and genetic diversity and transfers and all that stuff, but the ability, like, I know we're so excited, the tree kangaroo SSP, tree kangaroo, we are getting a tree kangaroo from Papua New Guinea. Its parents were
wild caught injured animals, and they stay in Papua New Guinea, but they had offspring, and that offspring was born at Port Moresby Nature Park. So we're getting fresh genetics into a very small population. And that's really cool and really exciting. And like, people are losing their dang minds over this, right? This is a huge deal for a population with like 42 individuals in the States, right? But like, also, we could just double the population or the genetic diversity by arguably collecting from, you know, other wild tree kangaroos as we catch them, as we do them for studies and stuff, and then just have that stuff without having to worry about the international transfers and the MOUs and all that stuff, right? Like, I know you still have to go through those things, but I would guess it's easier. Maybe I'm wrong, but I guess it's easier to get sperm than to have a country say, yeah, take one of our endangered animals, even if they are not able to be in the wild or whatever.
Right, exactly. You know? No, for sure. It generally does create less barriers, but then the biggest barrier, of course, is developing that technology. So that's why we're really lucky that we have so much work that's been done in cats over the years, because I think as a species or as a taxon, we're, I think, one of the most advanced groups with what we're able to achieve, which is really cool. So yeah, I mean, that's absolutely the model. Part of it is like working with different groups in the wild, making the sperm. I know that Bill, who I mentioned before, he's been to Mongolia and collecting like Pallas cat sperm, and, you know, from free living individuals. And, you know, that's a really small population as well. Here, like I think it was 11 founders is all that made up the Pallas cat SSP. And importantly, but then when we look at the genetic, you know, the relatedness and how much variation is there, the diversity is more as if there's only 4 individuals that made up the population, because not all founders are represented equally.
Sure.
And so, yeah, it's a great opportunity, right? Like, we don't typically go get animals out of the wild unless there's a reason they can't be there. So this is a great alternative to do that, where we, you know, if they're already being collected as parts of a study or, you know, things like that. So it's a really wonderful mechanism to kind of complement genetic diversity. I should say this too, it doesn't just have to go from the wild into our zoos, but it can actually work the other direction as well. I think Amur leopards are a great example of that. So critically endangered, there are at some point probably less than 20 left in the wild. Which I think is such a hard thing for us to think about as humans. Like, imagine if there were 20 humans left and that was it. Luckily, a lot of people rang the alarm bells. People kind of rallied around this species, and the numbers are 80, 100, maybe higher now, but their genetic diversity is just trash because there were so few of them. So we have more cats in zoos than there are in the wild, and our genetic diversity is better in zoos. So, and there are groups working on reintroduction efforts to take zoo-born animals that are proven, move them to this facility out in Russia, and then let them have cubs, but the cubs never interact with humans. And then sort of a soft release, a harder release, these animals going back into the wild, which is amazing. So yeah, I think I love that story, because we often talk about the importance of our diversity in zoos, but to what end? And that end, get more of it back out there. So
yeah, that is really cool. And I know, like, for a while back in the like 90s or whatever, SSPs were all like, oh yeah, we're a you know, an insurance population, so that you can do that. But then the AZA has moved away from talking about that, because we don't really, even when we do reintroductions, overall it's not through the SSP genetics necessarily, you know what I mean?
Yeah.
But it sounds like this is an example where it is, which is really cool, because I would like to see, it would be cool if we had the ability to do more movement that way, for sure. So that's really exciting.
Yeah, and I would say another cool story it's really topical in the U.S. are ocelots. So we have a big project right now working with Fish and Wildlife, with the East Foundation, with private Texas ranchers, kind of with that similar idea that Fish and Wildlife were already collecting these ocelots. So I should back up and say this. So ocelots, there's a small population in Texas. We don't know how many individuals, maybe 60. They historically had a much larger range, but due to habitat fragmentation and all of the things we're used to hearing about, they're a really small population. But what's cool is the ranchers are supportive of these efforts. They are actually pretty proud to have ocelots on their land. And unlike a jaguar, which they're not excited about, ocelots are not going to take their cattle down.
Right.
So it's this really cool group effort of, first we trained folks at Fish and Wildlife on how to collect sperm and freeze it with these cats. And then now we actually have a DV PhD, Ashley Reeves, that her PhD was in collaboration with us here at Crew and did a lot of training with her. So she's out there helping them, like trapping these cats, doing these health exams and collecting the sperm. What's really cool though, and so the idea is that sperm can be used in our zoos for AI. But the next step is they're actually building a rewilding facility down in Texas. So same kind of idea, taking these proven pairs, going down there, and then having these animals breed and releasing them back into the wild. And because they're building the support network between protected lands and then these private ranches, there's actually habitat for them to go into, which is really cool. So yeah, exciting. And again, the dream. It's like what we talk about what zoos do, but it's really cool when we, years and years and years down the road can actually make this stuff happen.
Yeah, that's awesome. That is very cool. So you work with cats.
I do.
And you said that as a taxa, they're far ahead of others. And is that because cats are cats are cats? Like, I kind of always talk about that. We just saw cheetahs playing with lures, and I've seen lions playing with the giant pickles and stuff like that. Is that why things are so far ahead, or are you just that smart?
Let's back up and just be clear. The we is the royal we, and people far before I even knew this was a career were working on this. So I would say as a whole, I think people who like cats are just smarter. So people who are working on cats are just going to get them further. But no, they are similar. They're actually very similar genetically. All cats evolved from a medium-sized ancestor roughly 11 million years ago, which is super short in talking about evolution time. So they're all closely related, but then they live all over the world. And so they're so adaptable. It's crazy to think about how close your cat is related to a jaguar or a lion. But like you said, you see the behaviors and you're like, oh, I can totally see these are the same. These are from the same individual. So they are similar, and we're lucky to have the domestic cat as a model. Although I would like to dispel the myth. Everyone thinks that domestic cats are so fertile. And it's really not entirely true. I think they're just really adaptable, and they're they can just live wherever. And so they do really well. Unfortunately, domestic cats living among us humans and living, hoboing our trash and all of that. They do really well alongside of us, but even domestic cats don't have great sperm. So, but what's cool about that is basically all that we've learned about the domestic cat for reproduction is almost entirely because of our interest in them as a model for wild cats. There actually aren't a lot of veterinarians that do assisted reproduction in domestic cats, although there's uses for that. We can sort of get into the ethics of breeding and this and that, but there are people who are ethically breeding animals and want to keep them, with whatever breed, keep the breed. And so there isn't a lot of support for them. But then cats actually are very similar to humans as well genetically, and not just in our genetic code, but how our genes are sort of organized. And so cats have, I think it's 250 naturally occurring mutations that cause disease that have analogous disease in humans.
Oh, wow.
And so, now there's all this CRISPR and genome editing stuff we can do. Back in the day, that didn't exist. And so these researchers interested in this particular mutation would go around to cat shows and do the cheek swabs and go run their magical PCR and find the mutation. And then they would bring them and begin a research colony. And so they could study how do we treat this disease so that they could find treatments for humans, which is really cool. So that's a whole other world, but it's useful to have assisted reproduction for these cats, because once that research is done, part of what we do is we work with universities. So then we go and, neuter the boys, spay the females, preserve the genetics, and then those cats can go get adopted, and they don't have to keep paying to keep this colony alive. But then down the road, they're like, oh, I got a grant. I want to study this. We need to resurrect the model is what they call it, which I think, it's Easter time. I think that feels very apropos. It's like resurrect the model. And I don't know why that phrase makes me giggle, so you need techniques like artificial insemination and IVF and all of those things to do that. So there is some use for domestic cat reproduction, but again, largely it's because of what we want to learn about the non-domestics. It's really helpful. It's why we have one of the reasons we have a domestic cat colony here. Because if you think, I've got this zany new idea how I could freeze sperm and it would live better, you don't want to do that for the first time in a lion.
Right.
So it's great to have that idea. But I will say this, and this should surprise nobody who likes cats. They're all very different too. There's unique issues with species and with individuals. And so there's a lot of variation. And so while we use the domestic cat as a model, there's still a lot of work on the other end of just figuring out, okay, how do I apply this? And certainly a lot of the work we do, it feels like a lot of the smaller cats are more closely related. And so they tend to be more similar. And then you just dial outwards from there. So it's really fun learning how to do, say, artificial insemination with a technique we developed in the domestic cats and then, worked a lot on in some of our smaller cat species. And it's like, okay, let's scale that up to a jaguar. And there's just a lot of individual variability that goes into it.
Very cool. Very interesting. So, especially with the bigger cats, you're very charming, but how the hell do you get sperm out of a cat?
Well, you say that. We actually are trying to work on some voluntary collection techniques, but we're not there yet. So the classic way is what's called electroejaculation, which kind of is exactly the way it sounds, but can be a little off-putting to hear at first. So first I'll say the cats are anesthetized and they are on a surgical plane of anesthesia. They are not consciously aware of what's happening to them. And so basically, you insert this probe into the rectum and there are kind of all the nerves that go in there. And I should say this too, ejaculation is a reflex like anything else. It's just a little different stimuli than when the doctor hits your knee to make you kick. Slightly different stimulus. So basically the nerves that relay that reflex back and forth are right in there, and nerves all talk to each other through electrical conduction. And so it's very low voltage. It's usually, I'd say typically even 5 or 6 volts would be the most we're applying. And it's in these sort of rhythmic up and downs. And that basically stimulates that reflex. And that's how we're able to get sperm. So that's the classic way. That is a challenge when you're talking about working in the field. You're thinking about being in Mongolia or the middle of nowhere, Texas. You may not have electricity. And so what's cool with cats, if you give them a particular type of anesthetic agent called an alpha-2 agonist, it actually makes the sperm move from inside the testis down through sort of the outflow tract. And it's in that last little section called the urethra. And so you can just pass a catheter in there and you get a sample. Now, you're probably not getting all of the sperm. We've done some studies looking at doing the catheter first and then following it up. So we're not getting all of it. And it's somewhat dose dependent, species differences, time dependent as well. Some species it works great in, some species it doesn't work great in. But that's a great way to get a sample. So it's nice, for example, if there's a cat at a zoo getting a health exam that I can't make it for, I can, communicate with that veterinarian of how to do this technique. And they have, we can access the sperm and look at it. So that's worked nicely. And I should say this too, for polar bears, electroejaculation does not work well. There's just a lot more anatomy to get through in a bear.
Sure.
And it just doesn't work very well. So that's pretty much the only reliable way you can collect semen from a bear species is with this technique. So it's nice. It's not just for cats.
Wow. That's really cool. Yeah. Yeah, I actually, when we got here, the first thing that I did, I signed in, and right next to me was an electroejaculation probe.
Yes,
and I was scooting back away a little bit just in case. I didn't know where this was going. I was like, God damn it, I'm here for an interview.
That one's going to maintenance. It needs to have a little repair. Well, it's funny, as you can imagine, these are not things that you go pick up at your local CVS, right?
Yeah.
Like they're just made by a handful of people, and these particular probes we have go back a very long way to an individual who I believe had his little station set up in his garage who would make these. And so, yeah, it's we keep them going as long as we can, but they need repairs.
Dude was the Steve Jobs of cat ejaculation. Yes. Who knew?
Yes. Yeah. So it's hard. And there are some commercial companies that are out there, especially more for like the bull side.
Mm-hmm.
And we actually are working with one of them, Lane is the name of the company, to come up with some probes that are like, because again, different sizes. So we have very different sizes. I think the one you had. One that you saw.
Check, please.
Let's be more careful with our language here. Okay. But that one is jaguar size.
Okay. Cool. Yeah, it wasn't that big. So
yeah, you should see the tiger.
Oh God. So yeah, so you talked a little bit about the domestic cat colony that is here, and some of why you have it. But like, I'm guessing that a whole lot of people are like, "Wait, what? They're just domestic cats at the crew building." So, can you explain that in more detail?
Absolutely.
And all of that?
Yes, we love talking about our cat colony. So I believe we have 45 cats in our colony right now, and so I should clarify too, because we call it a research colony. Some people, depending on their background, they hear colony, they're imagining the free roaming cats living outside. So these are housed individuals. They're not just running around the zoo. And again, their primary purpose historically was serving as a model for non-domestic cats. What we've become more interested in, though, or I should say, additionally, what we become interested in is thinking about studies we can do to help domestic cats for domestic cats' sake. And so one area that is more intuitive to our sort of background and skill sets is looking at what is a better way. We like to say a better way to spay. So I mentioned this earlier, right? We have this large number of free roaming cats living around not just the US, but the world. And people might hear other terms, feral, which is not necessarily correct. They're not all truly wild. A lot of them are very friendly to humans and might be only one or two generations from living in a home, or some of them are sadly just kicked out of their homes. Free roaming or alley cats, etc. So free roaming cat is one of the more preferred terms if you want to get really squishy. And I love this community cats to kind of reflect their spot. But yeah, we don't know how many. The numbers vary widely. 80 million roughly free roaming cats in the US and maybe 500 million worldwide.
Yikes.
And the way we've been addressing this is what's called trap, neuter, return, or TNR for short. And the idea is if you just take these cats out of the environment, more cats will just come and fill that niche. So instead, we're trapping them, neutering them, and then putting them back where they came from. So now they can't reproduce, but they're kind of guarding that area. So it works well in small scales, especially when there's some great examples from like islands, but it's really hard when it's an entire country. And these are grassroots efforts. The efforts vary by area. It's largely volunteers. And so it's just one of the biggest holdups, though, is having veterinarians and being able to perform these surgeries.
Yeah.
And then you have questions afterwards, like how long do I let them recover? Especially for the truly feral cats, is keeping them indoors for a week around humans so that their wound can heal. Is that really good for them versus letting them go and knowing that even under the best of circumstances, there's some rate of surgical complication? So there's a lot of challenges. It's not an easy, there's no easy answer. But where we come in is we're looking at ways to do this without having to do surgery. Just a single injection, a single treatment that would be effective. And we've done other various things over the years. We've looked at something called Gonacon, which is basically a vaccine against a reproductive hormone. But like other vaccines, it needs to be boosted, and it's something that's under USDA, and they've changed the formulation. And even though it works in a wide variety of species, the most recent formulation does not appear to be very effective in cats. And again, the challenges with you'd have to booster all these cats and et cetera. So we've worked with various groups over the years, and one of our collaborators at Mass General Hospital, Dr. David Pepin, developed a technique with gene therapy, which I know probably sounds scary to people, but this particular version is not messing with the DNA. It's not changing things. And sometimes that's what you need to do. There's a lot of human diseases that need that. But this is simply basically borrowing, I like to say, the machinery of the nucleus of a cell, which is what makes proteins. And it's just giving it a little job. It's like, hey, make this hormone, make a lot of this hormone. This is something the cats make naturally every month to regulate their cycle. So we're just going to make more of it. And it basically stops them from being able to reproduce. So we've published 2 papers on that. We know it works in adult female cats and kittens, which is really cool. Small studies, like 6 individuals, 7 individuals, but 100% effective.
Wow.
Put them in the room with males. They don't get pregnant. We have controls that do get pregnant, so we know that the boy knows what he's doing. Pretty cool. So that's still in the development stage. After we had our first paper come out, there was a lot of news, and then people started calling the zoo, hey, I've got some cats. Can I get a couple of those shots?
Oh, God.
And we're like, I would love to have that available, but it's everything we do has to get submitted back to the FDA, and we have to be very careful. And we want to do there's a lot more safety testing, just scaling it up to actually produce it for that many individuals. There's so many steps down the line. But what's cool about this is even though it only works in females, this technology could also work. We just changed the hormone. And I say that, we just changed the hormone. Okay, my expertise is on the reproduction side and the safety side. My expertise is not in producing the gene therapies, but for my smarty pants colleagues who that is their expertise, it really is, let's identify the next target. So I mentioned the Gonacon, it's vaccinating against GnRH, which is like the master hormone regulator. You could, in theory, use gene therapy to create that antibody rather than having the animal's immune system make it. You could then create that antibody. So they're basically vaccinated for life.
Right.
And that is something we're interested in and like looking to do, which is pretty exciting.
Yeah, that's really cool.
So it's exciting future. So that's the one area is sort of that contraceptive non-surgical side. But, and this is very apropos knowing that you're here for a behavior conference, is we realized domestic cats, they're not really that domestic, right? Like otherwise, all the other domestic animals you think about, humans domesticated them.
Right.
We made a decision and we're like, we need you to do X, Y, or Z. So we're going to breed you for that. Cats just showed up because humans are like, we should farm. And as we farmed and we stored things, the rodents showed up. So then the cat showed up. So it's kind of like the Chuck Norris joke that Chuck Norris doesn't get wet. He just allows water to be around him. Domestic cats did not get domesticated. They just allowed humans to be around them. And we do have a lot of behavioral issues with cats, and they are kind of wild. They do get the zoomies in the middle of the night. And what do? We take this wild animal and we give them their bowl of food and we give them everything they need, and they're bored out of their minds. So we're like, there's so many cats get returned to shelters for behavioral issues. You hear about, inappropriate urination and clawing things up. And so it's like, let's think about how can we improve cat behavior? And most of the behavior models we talk about are based on dog, on like sort of what we know in dogs. And so it makes perfect sense, especially thinking about it. We're at a zoo where we have so many tools about species we don't know a lot about. And how do we do preference assessments? How do we assess what they need? How do we assess if we're doing a good job? We're in the perfect place to ask that question, just with a species people don't think about, but probably needs it the most because we're forcing them to live with us.
Right.
So we do a lot of sort of behavioral type studies too. It's crazy. We just have simple questions. Do cats want the music on or off at night? It sounds so simple, but in a shelter, is it better to leave the music on so you're kind of controlling it, but then it's a constant sound versus off? So you don't have that constant sound, but you can't control the other sounds. Is that worse for them? But that's such a hard question to ask a cat.
Yeah.
And kind of before this, the way people looked at it, they would like play music with a speaker and then they would, I think, watch them for 3 minutes and note how much of the time they approached the speaker. Well, that doesn't really tell me. There was another study where the cats were under anesthesia and they played different music and they looked at like pupillary light reflex and jaw tone to see which one they liked the most. And I'm like, I don't think that would answer the question for me. So we kind of have to get creative. But what we did is we had cats basically like a double compartment type of cage. And I should say this, usually our cats are group housed, like running around. So we just had them in this cage overnight and they had an RFID collar. And the cage is set up the same, except when they walk to one side, it turns the music on. And when they walk to the other side, it turns off. And they figured it out so quickly.
Nice.
So we did this with several cats over several weeks. And what we learned is that they're just like us and they don't have a stable preference. Like sometimes they have it on all night. Sometimes, they never have it on all night. Sometimes they have it on for several hours. They're like, no, not today. Then they'll go back on. But it's really fun just to watch them like figure it out. Like you see them go over and it's on. Like almost all of them have this initial like back and forth. So we didn't really think we'd get like one single answer, and we're not surprised by it. But things like that we just try to ask questions for. So that's kind of the other fun use for our cats.
That is cool. And it brings up something that I struggle with a lot, which is like you were talking a little bit about how when you look at a study and a study gets done, people will maybe take it too far or maybe read too much into what they were seeing.
Yes.
And I see that a lot in the conservation side of things. And especially in the zoo conservation world, there was one study done recently where people were shown the same image of a cheetah, but one of them had like a hand and a collar like put on the photo, but it was the same photo because otherwise you couldn't control for like what the cheetah looked like,
Right.
And another one was with a keeper, and another one was just alone, and another one was they took the image of the cheetah but dropped it into like the its wild territory and stuff. And what do people think? And people said that the cheetah seemed less happy with the leash, and like some facilities have literally stopped doing cheetah runs or any encounters with leashes because of that one study. And I don't think that's a good enough study to say that is what effect that is having, especially since it was a still photo, and a photo that was photoshopped. And like, I don't know. But
I think it's tough. Yeah, I think in general, publications are hard. You've probably heard the phrase like publish or perish, and it's really big for sure in academia. We're a little bit insulated, but again, it's important for us to share our work as well. And we do a lot of grant writing, and they want to see, are you a contributor to the field? They're going to look at your publication history. And so I really want to start a journal called the Journal of Negative Results, because I think it's important. I think there's so much pressure that every study you do has to have this amazing conclusion. I actually I have it saved somewhere still, I'm sure. One of the first papers I wrote, and it was just basically back to this spermatogoneal stem cells, looking at different like antibodies, like basically staining what works, what doesn't. And that was all it was like, this worked, this didn't. I had this whole list of antibodies that I got, how I validated them, what I did to show that they didn't work. And one of my reviewers said, this paper knows exactly what it is. They're not overstating results. They're not trying to change the world. And they know that this paper's not, I think they said, is this paper going to change the world? Probably not. But it adds. And I just wish we could do more papers like that, where we just say, this was the idea. And even if we could be more open with, I wish I'd done this differently.
Right.
You know, like, get it out there. You've done this, but here's what we need to think about. But there's so much pressure from institutions and from, like, depending on your reviewer, to make it this big thing.
Right.
This has to be this huge conclusion that everybody's going to use from now on. And half the time people cite these papers, they don't even read them.
Yeah.
It's crazy. I go back and sometimes and I'll you're trying to find that original citation. I should also mention I have a background. I did a lot of debate in high school. So I'm really big on, like, I want the original source.
Right, same.
I go back and I want to know. And then you there's this citation you see all the time and you have to go back. And then this paper cites this paper and you finally find the original paper. And I'm like, this doesn't say any of this.
Yep.
But nobody goes back and reads it. So it's definitely a challenge with publishing that is across the board. And yeah, I agree with you.
Yeah, it's interesting. And the thing that drives me crazy about it is being in this whole world, not just the sciencey side of it, but like I will then hear, like I went to the tree kangaroo workshop and half of the presentations were on things that failed. Because they learn from that, right? And what they learned from the failure, and other people who tried the same stuff would speak up and go, oh, yeah, it didn't work for me. Oh, it did work for me. So maybe that's an individual thing, and it's not sciencey. You can't publish it. It's anecdotal. It's based on what these people saw as they were doing their jobs, not like in a scientific study. But everybody learns from it. And it makes the point, and nobody's ashamed to, same here with the behavior stuff at, ABMA and IAATE. People talk about what works, what doesn't work. There are videos that we watched this week of people being like, and then we tried this, and you hear half the room go, ooh, and they're like, and obviously that's not what we were going for. But that matters. That's important. I think sometimes some of the trainers learn more stuff seeing what not to do.
Oh, for sure.
That, but yeah, obviously to get published, you can't just say, well, this paper didn't work. Please publish it.
But I mean, I think I talk about this too when we're trying to do something. This was really good advice from my PhD advisor, Carol Kiefer, that is like, if you are going to show something that didn't work, how, then you need to think about your experiment, not from, I'm trying to make this work. If you're really going to flip and say, I don't think this doesn't work, why doesn't it work? And can you show that?
Right.
So like actually doing the exhausting all sort of endpoints of, I did these 20 things, so this doesn't work for this. And to me, that is valuable information. It's hard to find the right journal to let you publish that, to be interested in it, but you certainly can. And I think the more we would do that, again, that's why I want my journal of negative results.
Right, I like it. Yeah.
But it would be very rigorous. And we're not just going to be like, I don't care that you tried it this one time. That's not publishable. But show me all the different ways. Tell me what you thought about. Tell me your theory of why this, that, the other worked. And tell me if there's anything else you could do. Exhaust that. Write that up. How many people, I wonder, have done the same stupid experiment over and over again at all these different institutions because they didn't know that someone else already tried and it didn't work.
Yeah, definitely. It's interesting. It's really interesting. So is there anything else you want to say about the stuff that you're doing here or anything really?
That's, again, convoluted answer. We'll be here the rest of the day. No, I feel like you've asked me really good questions. There's nothing that really is screaming to me that I need to say out loud.
Cool. Great. And then how can people support Crew?
Well, you can actually, we have our website, so you can actually Google, I think it's Imperiled Cat Signature Project, or just if you Crew Cincinnati Zoo, we actually do have a link on there to donate. And, we are very driven by grants, and it really determines how much we can do, all this travel, going to these zoos, doing all this support. It does cost us a lot of money. So help us, and we'll keep doing it.
Very cool. Love it.
It's time now, don't you know? We've come to the end of the show. But there's one tale left to go. You're gonna laugh and say, oh no. It's time for the Rossifari poop story.
Oh my gosh. Okay. So, semen collecting a jaguar, as one naturally does. And they will, again, it's electricity, so they will react. And a lot of times we're actually looking for good hind end extension. And this particular jaguar, his tail got into the game, and it was like the Pink Panther, like on my face. And it was just the most endearing thing. I felt so connected to this cat. So my boss at the time, Bill Swanson, was like, get that thing out of your eye, or, you're going to get pink eye from that. And I'm like, stop it. Don't you dare stop what we're doing here. This is between me and Bebu. Stop it. So I'm not even kidding you. We're on this like 2-week Texas road trip. We're doing it like going to a lot of zoos. It was really nice for a while. We had everybody on the schedule where they would schedule their annual exam for their cats like in the same time frame. So we could just do this loop around Texas. So I'm at another zoo. We've semen collected, I believe, 2 jaguars. I've got my sperm. I'm showing everybody on the microscope slide. So everyone's looking at this microscope, and I'm packing up to leave. And this is like, I remember this brief, like so clearly. This is just when Subway started putting guacamole on your sandwiches for the first time. And I was like, yes. So I'm eating my Subway sandwich and I'm cleaning up. And then I'm like, how did I get guac on my face? And I'm like, okay, I am working with probes that go in butts. I should not be eating while I'm cleaning all this up. And I'm cleaning up more, and I'm like, I do it again. And I'm like, how did I get avocado on my face twice? I put the sandwich away, and then I'm like, that's not avocado.
Oh no.
And all these people have looked in this microscope, and I'm like, oh dear. So I did confess to the veterinarian at that zoo. I said, please wait until I leave. But please tell everybody I'm so sorry, and everyone's about to get pink eye.
Oh no.
But the best part of that is I never even told my boss I had pink eye. I was randomly started on the right side, and he was driving, and we had like a 6-hour drive, and I'm just like subtly wiping it out. He's like he has, I wouldn't say a germaphobe, but he probably would have sent me on a plane back home. He'd have been like, you're not here anymore. So I didn't want to tell him. So I did like a telemedicine call and took an Uber to the pharmacy to pick up my eye drops. And I'm so glad I did, because then in the morning, my eyes had already like gotten so gross. And then I'm in a hotel room I don't recognize. And I don't know the room, and my eyes are so gross. I can't open them.
Oh no.
So I'm trying to get to like a washcloth to get wet, but I don't remember the room because I just got there.
Oh yeah.
And so I made it to the front door like 4 times before I made it to the bathroom. So finally did. Mind you, now I've touched the entire room with my pink eye hands. And so after I can see again, I write this note to like the people, but I didn't want Bill to see it. So I was like, where do I put it where they'll walk in and see it? So it's on the floor, and it's like, I'm so sorry. This room is contaminated with pink eye. Please don't touch anything.
Oh my God.
Heart room 409 or whatever. So the best part is, of all of this, I really thought it was the jaguar. And then I'm telling my friend here at Crew when I get back about it, and she is making this face at me like I'm the most, like she has this just look of horror. And I'm like, I know it's pink eye. It's fine. Like people get over it. And she's like, no, her whole house had pink eye. And I was over at her house like a week or 2 before that. Her kiddos had pink eye. I just kept my immune system just fought it for like 19 days. I think I'm like a world record holder. So I actually got it then. It was not from the jaguar, but yes,
that's amazing.
I thought I got pink eye from a jaguar tail, and I actually thought that was pretty cool.
Amazing.
Just from kids.
So good.
Yeah.
So good. Well, thank you for taking the time to do this.
Of course. It was a pleasure. I'm glad it finally worked.
Woo. All right, so there you have it, folks. I'm always so grateful to the Cincinnati Zoo for being so willing to partner with Rossifari, especially because they have their own very successful, zoo podcast. And I think it's really cool that they have never seen this as anything but another opportunity to, be good people and help out and get the word out there and all the things. So I have loved the Cincinnati Zoo since before I went there, and definitely since the first time I went there. And I appreciate how much they have been a part of Rossifari. So yeah, and how great was Dr. VanSandt, right? Like coolest human. Loved it so much. I hope y'all enjoyed that one even half as much as I did, because my goodness, I loved it a lot. So yeah, I will be back here on Friday with a brand new episode of Rossifari Zoo News. And yes, my plan is to actually do an actual episode of Rossifari Zoo News this week, because I'm not moving this week. So there's that. Before I get going, I do want to take a moment to say thank you to all of my patrons, especially my Red Panda level patrons, Dr. Laura Shank, Dr. Stephen Williamson, Barbara Bennett, Jenny Owens, Melissa Reed, Carrie Kirkpatrick, John Lewis, who is also here for stickers, Kevin Williams, and Cormac McMuffin. Meow. And until we touch base again, remember, friends, the word credits backwards is Steiderk. The Rossifari podcast is produced, hosted, and engineered by John Rossi. Editing and fact-checking by John and Dr. Zoe Rossi. Our theme song is Sevens by Nathan Burke, performed by Nathan and John. Interrupting John theme and additional voices by Taylor Isaac Gray. You can reach John directly on Instagram and Facebook at Rossifari or by email at rossifaripod@gmail.com. Rossifari is part of the Daydreamer Media Network. Now, stop listening to me and go visit a zoo.